and please take it away.
Okay. So the research title is Sinomenine inhibits amyloid beta-induced astrocytic activation and protects neuron against indirect toxicity. So neurodegenerative diseases like Alzheimer's are uh, prevailing diseases that cause the cognitive dysfunction, memory impairment, and about 40 million people today are affected by the Alzheimer's disease that are posing uh, economic and the social burden on the society. It also ruin the functional independence of the people of the society. Therefore, it is needed to present or produce some therapeutic interventions to fight against the neurodegeneration. So, through my research study, I have found that uh, the amyloid beta, that is a pathological hallmark of the Alzheimer's disease, uh, is are getting inhibited. Its toxic effect are getting inhibited by a drug which is called the Sinomenine, which is recognized in China as a therapeutic medicine for the rheumatoid arthritis. So, first of all, amyloid beta, which is produced by a proteolytic cleavage of the amyloid precursor protein, they deposit extracellularly and cause the toxicity via uh, neuroinflammation by causing the neuronal cell death. So, first of all, we have checked the neuronal health by seeing the or visual by investigating the effect of the amyloid beta on the neuronal hippocampal cell, that is the ST22. And we have found that the amyloid beta, which I have referred here, ADDL, this amyloid beta, when we treated the ST22 cells that are the neuronal hippocampal cell with the ADDL, it causes the increase in the production of the ROS. While first we have checked with the LPS whether that our system are working well or not. In the LPS also we have found that ROS is increased. Further, when we have seen the toxic effect of the ADDL and ROS, and we have found, yes, our system are working well, that our ST22 cells are responding to the toxic species, then we have explored the effect of the Sinomenine. So, Sinomenine, which is in the China, were used as a medicine for the rheumatoid arthritis and having the neuroinflammatory effect, anti-neuroinflammatory effect. So we have seen its effect. So we have found that that increase in the production of the ROS were decreased by the Sinomenine. And when we have checked the nitric oxide level, another the toxic species, and we have found the same, that yes, ADDL has increased the level of the production of the nitric oxide while the Sinomenine has decreased it. Further, we have explored whether the Sinomenine have effect on the cytokine production on the amyloid beta treated hippocampus cell. And we have found that when we have measured the series of the cytokines, such as the IL-12, IL-6, TNF, IFN gamma, IL-10, and the MCPs and all, we have found that amyloid beta have increased the level of the all the cytokines, which include the anti-inflammatory cytokines too, that are the IL-10. And Sinomenine has decreased this cytokine level. So from our study, we have found that Sinomenine are here decreasing the level of the cytokines in the cytokines and the toxic species. In the case of the, sorry, I have mentioned one thing wrong here, that because here we are treating the astrocytic cells with the amyloid beta, and we are seeing the effect of the Sinomenine against amyloid beta treated astrocytic cells, not neuronal hippocampus cell. So this is because we want to see the effect of the Sinomenine against amyloid astrocytic activation that are mediated by the amyloid beta on the neuronal health. So till here, we have found that the amyloid beta has caused the astrocytic activation that are uh, indicated by the increase in the level of the ROS and the NO, how, and also in the increase of the cytokines, which shows that amyloid beta are increasing the toxic species in the case of the in astrocytic cells. While when we have treated our astrocytic cells that are further treated with the ADDL, amyloid beta, with the Sinomenine, we have found that Sinomenine have decreases all these toxic species. Then we thought, and we have explored further, that what is the effect of the astrocytic activation on the neuronal health. For this, we have treated the conditioned media of the astrocytic cells that are treated with the amyloid beta and that are treated with the amyloid beta and Sinomenine. So when we have treated the conditioned media obtained from the activated astrocytes and the astrocytes that were treated with the Sinomenine, we have found that in the case, first we have done with the cell lines, and we have chosen the hippocampal neuronal cell line, that is ST22, and we have found that astrocytic condition media uh, causes activate the amyloid beta treated astrocytic condition media, where have increased the number of positive cells. The tunnel and ASA are basically used to see the apoptotic cell death in the case of apoptotic cell death. And we have used this tunnel assay, and we have found that astrocytic activated astrocyte condition media have increased the number of the tunnel positive cells. That shows the number of neurons are dying. While when we have treated with the Sinomenine plus ADDL condition media that are obtained from the astrocyte, the level of the tunnel positive cells were less. So which shows that Sinomenine here is protecting the neurons against the astrocytic activation. So for more validation, we have used the human neurons and we have performed the same experiment and all. And we have found that in the case of the human neurons also, the Sinomenine has protected the neurons against the amyloid beta induced astrocytic activation. So from our study, it has shown that the amyloid beta is protecting the neurons against the astrocytic activation, which are mediated by the amyloid beta. And further studies are required to see its mechanism and all, although we have not explored it. But yes, it is required to know more about the therapeutic effect of the Sinomenine against astrocytic activation or in protecting the neurons against cell death.